everyone, I am Angela Amalgan, and in this video I will be discuss about the introduction of liquid chromatography and mass spectrometry. So before we proceed, let's talk up first what is the introduction of this topic. So liquid chromatography and mass spectrometry, it is a component of an analytical chemistry technique that combines the separation of capabilities of liquid chromatography or the high performance liquid chromatography or HPLC with the mass analysis capabilities of the mass spectrometry or the MS. Liquid chromatography separates mixtures with the multiple components. Mass spectrometry provides a structural identity of the individual components with a high molecular specificity and detection of sensitivity. This is the example of these components is that we use is in a bicycle. The basic bicycle technique can be used to analyze biochemical organic and inorganic compounds commonly found in a complex samples of environmental and biological origin. Therefore, the LCMS may be applied in a wide range of sectors including biotechnology, environment and monitoring, food processing, and the pharmaceutical, agrochemical, and cosmetic industries. Later on, I will be discuss that one by one in this video. So here are the objective for today. First is to discuss the difference of application of the liquid chromatography and mass spectrometry. Second is explain what are the importance of liquid chromatography and the mass spectrometry, or the difference component of the difference components. Then lastly is identify the principles of HPLC. So first application of these components is the coupling of MSLC system is attractive because liquid chromatography can separate delicate and complex natural mixtures which chemical composition needs to be well established. The example of this is a biological fluids, environmental sample, and drugs. Secondly is LCMS has become one of the most widely used chemical analysis techniques because more than 85% of natural chemical compounds are polar and the thermally level GCMS cannot process these samples. As an example of high performance liquid chromatography or the mass spectrometry is regarded as the leading of the analytical techniques for proteomics and pharmaceutical laboratories. Next application is in the analysis of food. In analysis of food, you can use this component in food and analyze a pesticide in order to killing bacteria or then to prevent the seizure of the health. And then lastly is the plant phenols. Fourth one is the pharmacokinetics. Pharmacokinetics is the LCMS widely used in the field of bioanalysis and is especially involved in the pharmacokinetics studies of pharmaceuticals. Then pharmacokinetics studies about the drugs are needed to determine how quickly a drug will be cleared from the body organs and the hepatic blood flow. And the mass spectrometry is analyzer are used in these studies because of their shorter analysis time and the higher sensitivity and specificity compared to UV detector commonly attached to high performance liquid chromatography or the HPLC system. Then the fifth one is the proteomics. It is used as a method to detect and identify the components of a complex mixture. The next one is the metabolics. Lastly is the drug development. So under the drug development, it is also to develop the peptide mapping, um, then the glycoprotein mapping, natural product replication, bioaffinity screening, in vivo drug screening, metabolic stability screening, metabolites identification, impurity identification, and quality control. So, according to my research, liquid chromatography and mass spectrometry is the powerful analytical techniques used for separation, identification, and quantification of both unknown and known compounds, as well as to elucidate the structure and chemical properties of different molecules. So, what is the difference of liquid chromatography and mass spectrography? So, liquid chromatography and mass spectrometry or spectrography is the technique which perform separation by liquid chromatography and mass analysis with the help of the mass spectrometry. It is also called an electrospray needle. Is used as a bridge to connect the liquid chromatography with that of the mass. Then next one is there are three main parts of the liquid chromatography and the mass spectrometry. First is the chromatography. From the word chromatograph is the instrument used to conduct chromatograph. The purpose of this is to analyze the any drugs. Is to confirm the identity of the drugs. Also to know the names of the drug, and provide the quantitative result, and also monitor the progress of the therapy of a disease. Next is the interface. Interface um, it chromatography also is a liquid of the chromatography separation is performed, which is detected with the help of the photodiode array components, then transferred to the interface. Then the second one is after the chromatography, the second transferred to the interface. An interface is it is a liquid volatilized. So siya na yung mag-transfer to the mass spectrometry. Then after that, pagkatapos niya ma-transfer, si spectrometry na naman ang tatanggap na niyan. Then with the help of that is various ionization techniques. The compounds is ionized and then it is analyzed by mass analyzer. The various mass analyzer are used to the quadrupoles and quadruple ion traps. Time to flight, or this would we call TOF, or the time to flight reflection, or the TOFR, and the ion cyclotron resonance, or the ICR, and the mass analyzer. So another important things is the liquid chromatography is a method of the physical separation in which the components of liquid mixtures and distribute between two immiscible phases. The example of this is the stationary and mobile. So the practice of liquid chromatography can be divided into 5 categories, which is these 5 categories. First is the absorption of the chromatography, from the word of absorption. Absorption, from the word of absorption, it means you can absorb the, what do you call that, the example. And then the second one is a partition chromatography. Then third one is the ion exchange chromatography, and then the size exclusion chromatography. In the size exclusion is it's focused in the size. Then lastly is the affinity of chromatography. Then among of these, the most widely used variant is the reverse phase mode of the partition chromatography techniques, which makes use of a nonpolar hydrophobic stationary phases and the nonpolar mobile phase. The mobile phase is a mixture of water and the other polar solvents. The example of this is a methanol and the isopropanol and the isonitrile. The stationary matrix is prepared by attaching in the long chain alkyl groups. Example of this is octadiesel. Then to the surface of the irregularly or the spherical shape of 5 diameter silica particles. So let's proceed to the principles of SPLC. But before that, SPLC is from the word um, um, of HPLC, the high performance liquid chromatograph. It is also the instrument used to conduct the chromatograph. So, first principle is that typically of the sample of the interest are injected into the mobile phase stream delivered by a high pressure pump. Then second one is the mobile phase containing the analyzed permits through the stationary phase bed in the definite direction. Then the components of the mixture are separate depending on their chemical affinity with the mobile and the stationary phases. Next one is the liquid solvents or the mobile phase is delivered under the high pressure into the packed column containing the stationary phase. Then next one is the high pressure is necessary to achieve a constant flow rate or the reproducible chromatograph experiments. Next one is depending on the partitioning between the mobile and the stationary phases. The components of the sample will flow out of the column at a different times. Then next one is the column is the most important component of the liquid chromatograph system and the design to withstand the high pressure of the liquid. So, so that would be all about the principles of the HPLC. So, from the summary, is the liquid chromatography is a separate techniques in which a mobile phase is a liquid where sample ions or molecules are dissolved. It is carried out either in a column or plane. Mass spectrometry is an analytical tool used in measuring and the mass to change ratio of the more molecules present in the sample. This measurements can often be used to calculate the exact molecular weight of the sample components as well. So this component, they call this component is a technique to conduct the chromatograph. So I think that would be all this about my presentation. That's all, and thank you.